So I want to return to this figure quickly before we move on. A good analogy or a good example to help illustrate this concept of CO2 moving out, pulling substrates through the carbonic anhydrase reaction, or adding CO2 and pushing through to the other direction, is to think about a giant pot of soup or a punch bowl full of punch, bowl full of water, something like that. The level of the water, the level of the soup is consistent. And if you take a ladle of water out, you're not left with a giant divot in the surface of the water, because the rest of the water will fill in that hole. Similarly, you add a ladle of water or a scoop of water on top, and it's not left as a mound on top of the surface, it spreads out so the surface is even. And the same thing happens with CO2 here. If you remove CO2, you're not left with this low concentration of CO2. Bicarbonate and protons combine to fill in the gap. And if you add CO2, you're not left with this high concentration of CO2. It splits up into bicarbonate and protons to even out, maintain a level, equal playing surface, so to speak. And we're doing that at the lungs. We're ladling out CO2, scoop by scoop. It's being dissipated to the environment. And so we're balancing out that equation by combining bicarbonate and protons to even out the level of the water. And at the tissues, we're adding scoops of CO2. So a nice little analogy to help understand the near equilibrium nature of this equation. And maybe that helps to understand that removing CO2 pulls these items through the equation, or adding CO2 pushes the equation in the opposite direction. So important concept. Also review how these are somewhat related. That is the movement of CO2 and the movement of O2. But we are moving on to control of respiration. We breathe regularly. Why do we breathe regularly, and why does breathing increase in situations like exercise? What controls ventilation? And I mean ventilation, even though it says respiration here. Remember, we're using respiration to describe cellular respiration. What controls ventilation? Ventilation, like we know from the start of this chapter, occurs because of contraction and relaxation of the ventilatory muscle. So those things have to be controlled. Those are the things that are turned on or turned off in response to various stimuli. And just like there was a cardiovascular center in the brain that controlled heart rate, there's a respiratory center in the brain that's closely associated that controls breathing rate and breathing depth, for that matter. There are two shown here, really two, and then one of them is divided in half, and we're focused on the dorsal and ventral respiratory groups of the medullary respiratory center. That's the most important one for normal breathing, as we're going to discuss it. And in brief, general overview type terms, the dorsal respiratory group is responsible for normal breathing, normal function. It sets normal pace and normal depth. Is it just me, or is this flickering a lot more than usual?
Yeah.
So after our first midterm, I went to IT services to see if they had any other adapters. They had none on hand, and they said they were going to order them. And I followed up yesterday, and they hadn't put the order in yet. So that's just a little inconvenient. Let me just try one of these other ports. Fingers crossed. Okay. Where were we? The dorsal respiratory group controls normal quiet breathing. Normal pace, 16 breaths per minute. Normal depth, 500 mils per breath. And you know this from the lab when you did resting tidal volume measurements. Normal quiet breathing, light rhythmic activation of the diaphragm and the external intercostals is what activates inspiration. And then expiration is relaxation of those muscles, turning off of those muscles. There's no forced expiration in this normal quiet breathing. The dorsal respiratory group says activate, and the diaphragm drops, the external intercostals raise, the volume of the thorax increases and air moves in. And the dorsal respiratory group stops saying contract. That's when everything returns to normal and air is pushed out. So activation of the dorsal respiratory group brings air in. Deactivation pushes air out during normal quiet breathing. But breathing's not always normal and quiet. If you're stressed, if you're anxious, if you're exercising, breathing rate and breathing depth increase. It becomes more forceful. As I stand here and lecture and have to breathe around the words that I'm saying and push air out more forcefully and bring it in forcefully too, I have to activate other accessory muscles to do that. Those are controlled by the ventral respiratory group. The ventral respiratory group is really what coordinates any extra breathing required. It can turn on the normal muscles more, so the diaphragm and the external intercostals. It can accentuate their involvement. And it will also turn on other muscles like some neck muscles, pec minor, all of the accessory muscles that would squeeze any extra space or volume out of the thoracic cavity. So forceful inhalation by activating neck muscles, pec minor, the normal muscles of breathing more, and forced exhalation by contraction of things like the abdominal muscle group, squeezing and pushing air out, the internal and innermost intercostals, contracting and decreasing the volume of the thorax. So forced inhalation and exhalation controlled by the ventral respiratory group. Now it's worth mentioning, we've only talked about two of the main parts of the medullary respiratory center. The pre-Boatzinger group is analogous to the SA node. This is thought to be the thing that sets the pace. Like the heart's SA node would set the cardiac rhythm. This center controls the pace. It says when to activate the muscles. And these two groups relay that message and tell those muscles how forcefully to activate. So the pace is thought to be set by the pre-Boatzinger complex. If that's how you say it, I don't know. So important distinction between those two. Dorsal respiratory group always active and then can involve the ventral respiratory group if required. So this is a schematic flowchart that outlines everything that we've just said. On the left-hand side, the dorsal respiratory group that's active during quiet, normal breathing. Inhalation, when it's activated, 2 seconds-ish of activation, brings air in during inspiration, and then those muscles are deactivated for 2 to 3 seconds, and passive elastic recoil pushes air out. And you know the mechanics of this already. The rib cage falls, the central tendon of the diaphragm pulls it upwards, the elastic recoil of the lungs returns them to their normal non-inflated shape. surface tension in the alveoli, want to collapse the alveoli. These are all the things that return the lungs to their normal shape, pushing air out. On the right-hand side, with forceful inhalation and exhalation, notice the dorsal respiratory group is involved, and it recruits the ventral group to activate inspiration and expiration more forcefully. So it's always involved, but it augments the signal. If you follow the flow chart through, you see a larger engagement of the normal respiratory muscles, and now you start to see things like sternocleidomastoid, which is a muscle in the neck, scalene muscles, which we referred to briefly, pec minor, that are activated to increase thoracic volume during forceful inhalation. And then the abdominal muscles, obliques, transverse, rectus abdominis, that contract to squeeze the abdominal cavity, which increases the pressure in the thoracic cavity and pushes air out. So these centers tell the muscle to activate or not, and how forcefully to activate or not. But what makes these signals initiate, or these centers initiate those signals? What's being monitored that prompts a change? Did you all get that down? I'm sure you did. Okay. What's being monitored to prompt a change in the firing of the dorsal and ventral respiratory groups? Chemoreceptors, largely at play here. Now, so far everything we've talked about is pretty unconscious. If you sit here breathing normally, quietly, you're not in control of your breathing, although you can take control at any time. When you move to exercise, the workload sets the demand. And you can't really cheat. You can't breathe more than you need to or less than you need to. The forced inspiration and expiration are set. But you can control breathing whenever needed because there are cortical inputs. The cortex can control those ventilatory muscles. And now that I mention it, you're probably thinking about it, and it's hard to let them go sometimes when you have conscious control. But even though you can consciously control those muscles, they can be overridden by the chemoreceptors that are constantly sensing composition of fluids in the body. Cortical influences can be overridden. So when a child says, I'm going to hold my breath and pass out, if you're ever babysitting or if you end up having kids and they're trying to get their way, you say, fine, go ahead. It's impossible. You can't hold your breath long enough that you pass out or die. Or maybe if you pass out, normal breathing resumes as soon as you do. They can't voluntarily hold their breath because that signal gets overridden by the chemoreceptors. So the signals are chemical, and they can override the cortical influences on the dorsal respiratory group, or either respiratory group. And it's always monitoring and sending information on blood gas concentrations, O2 and CO2. Because that's the point of breathing, right? We are trying to maintain appropriate levels of these gases in the blood. Adequate oxygen saturation. We don't want too much CO2. And by association, through the carbonic anhydrase reaction, we don't want too much of a high hydrogen ion concentration floating around. So we have two groups of chemoreceptors, what we call central chemoreceptors and peripheral chemoreceptors, and they both relay information to the respiratory centers. The central are a little new. They are in the medulla, so they're right next to the medullary respiratory center. which is probably pretty handy to have a local sense of the composition of fluids. And these central chemoreceptors directly monitor the cerebrospinal fluid. And there's actually a very similar composition in the cerebrospinal fluid and in the blood. So it's effectively measuring the blood. The peripheral chemoreceptors are in those spots where we saw baroreceptors in the blood pressure section. There's clusters of cells in the aortic arch and in the carotid arteries. Aortic bodies or carotid bodies. Now, all told, altogether, they monitor these three elements: PO2, PCO2, and the hydrogen ion concentration. Remember, these latter two, PCO2 and hydrogen ion concentration, or pH, same thing, are very tightly related through carbonic anhydrase reaction. So we want to control PCO2, but the actual signal that's being registered is a change in pH. Doesn't matter, they're both very tightly regulated or related. So all three of these elements are controlled, but not all chemoreceptors sense all three. Not all chemoreceptors sense all three. So central chemoreceptors in the medulla, picture it right here in the brainstem, medulla oblongata. We have central chemoreceptors there, and they respond to changes only in PCO2 and pH. They don't monitor O2. Central chemoreceptors, which are arguably the most important, that provide the most potent signal, only sense PCO2 and or pH. The peripheral chemoreceptors, on the other hand, sense POC2, apparently. It's a new fun gas. They sense PCO2, pH, and PO2. So the peripheral chemoreceptors cover all the bases. And I think it's also important, as we get into some finer details, to note these are sensing the composition in arterial blood, or on the arterial side. So carotid arteries, aortic arch, obviously arterial fluids, right? Arterial blood. CSF, very close in composition to the arterial blood. There's a very easy exchange between the CSF and the blood. And if you remember way back when, we talked about that in the blood flow and the CSF production in the brain, I think in week 2 of semester 1. So these are on the arterial side, and I make that point because that will inform what normal levels we should look for. Normal PCO2 and normal O2. When is ventilation stimulated? If PCO2 ever gets above 40 millimeters of mercury, the central chemoreceptors snap into action. So you can see why it's important to make that distinction. On the venous side, PCO2 is in the 45 millimeters of mercury range. On the arterial side, we limit it to 40, and we want no more than 40. 41, you breathe more heavily, due to the influence of the central chemoreceptors sensing that small change. And if I were to put arbitrary percents on the relative involvement of these two groups, I'd say about 80% of the drive to breathe comes from this avenue. Small increases in CO2 on the arterial side. Major response shown here. That can be modified slightly if there's also the stress of low PO2. The peripheral chemoreceptors will also respond to a higher CO2 and a higher hydrogen ion content, or a low pH. But they will increase their involvement if PO2 falls dangerously low. Now remember, on the arterial side, PO2 is pretty much 100 millimeters of mercury. Right? Of course, you remember that. You might want to remember that for Friday, next week, when we have our second midterm. So PO2 on the arterial side, about 100 millimeters of mercury. These are only active, the peripheral chemoreceptors only add to this signal when PO2 drops below about 50 or 60 millimeters of mercury. That is, we allow a lot of error or a lot of movement in PO2 and very little movement or variation in PCO2. Ventilation is much more sensitive to changes in PCO2 than to changes in PO2. Which might be counterintuitive, because oxygen is so vital to maintain normal body processes. Wouldn't that be the most important blood gas to maintain? It is important. It's not unimportant. Maybe you're thinking back, and rightly so, to the hemoglobin association curve. At a PO2 of 60, we're still 95% saturated. We're not in any danger of having a low oxygen delivery to the tissue, so that's fine. But a high PCO2 will really acidify body fluids, and that is dangerous for normal body function. We want to prevent that at all costs. The body becomes acidic, proteins stop working, things break down. Not a good situation. So we're very sensitive to changes in PCO2. So, all told, these things work properly and we maintain normal oxygen and CO2. Conditions called normoxia, or what you might refer to as normocapnia. But if blood gases ever stray too far from their normal concentrations, we can observe changes in CO2 or changes in O2. Just focus on the first set of bullets initially. If CO2 is artificially high, it's a situation called hypercapnia. Hyper being higher than. Capnia, I'm not exactly sure how that relates to CO2. But hypercapnia indicates a high PCO2. This is the thing that is registered by the central chemoreceptors. Hypercapnia makes ventilation ramp up right away. Hypocapnia is also possible. You'll see this if you've ever hyperventilated or seen someone hyperventilating. You're blowing off a lot of CO2. PCO2 on the arterial side will drop. And doing that actually really lets you hold your breath for a long time. You can double or triple your breath hold time by hyperventilating for a minute and then trying to hold your breath. PCO2 drops, and if you're most sensitive to PCO2, you've got a lot of reserve capacity to increase PCO2 before you trigger breathing. That's also why when you see someone hyperventilating, they'll put a paper bag over their mouth to stop them from losing so much CO2, because having too much is bad and the body becomes acidic. Having too little is also bad and the body fluids become more basic. You want to maintain that within a tight range. Now, it's generally not possible to affect one and not the other. So if you are hypercapnic, you might also be hypoxic. A low O2 or low PO2 on the arterial side describes a low oxygen content, which is dangerous because the tissues do need oxygen, of course. It is vital. experience this a lot at altitude, oxygen deficiency, low arterial PO2, resulting from any deficit in function that gets oxygen from the lungs to the body, or lungs to the tissues. Airway obstruction, altitude, etc. And we allow a certain amount of hypoxia. You can survive in hypoxic conditions. Your brain starts to go a bit loopy. We do allow some of it. Hyperoxia, on the other hand, is really hard to initiate, to instigate. Because hemoglobin is near fully saturated under normal conditions. Hyperoxia would mean higher PO2 than normal, and it's really hard to have a higher PO2 than normal. Simply because hemoglobin is so good at binding oxygen. That said, you can squeeze out a couple percent if you're a high-level athlete and really want to perform well. Hyperoxia can help you in that regard. So, ventilation is controlled to maintain blood gases within acceptable ranges and prevent these wild variations. We are much more responsive and sensitive to changes in PCO2 and changes in O2. Hypercapnia, hypocapnia is what we want to limit. Hypoxia we can live with for a little bit. So we've covered all of these points, all of these unimaginative bullet points. We've talked about the anatomy of the respiratory system. You know full well how oxygen is bound and transported in the blood, and we've articulated the process of gas exchange. And it is the exchange of gases, loading and unloading of gases at different areas in the body. So let's summarize. Lots of information in this chapter. Movement of air only due to activation and relaxation of the ventilatory muscles. It's all about changing the volume in the thoracic cavity. And changing that volume will impact pressure in the airways, which prompts air to move. Higher volume, lower pressure. Lower volume, higher pressure. And so manipulating pressure in the airways prompts air to move according to its pressure gradient. Air moves according to its pressure gradient. Air contains multiple constituent gases. If a gas moves, it moves according to its partial pressure gradient. The air that we're breathing now is a mix of at least 3 main gases. There are others in trace amounts. The 3 main gases that we're concerned with, we talked about, and we know what their partial pressure should be. And if we can establish a gradient, you can understand how those individual gases will move into or out of the body. Now, oxygen being the candidate gas that we use in our examples will bind to red blood cells, specifically to hemoglobin, containing 4 heme molecules arranged in a nice square pattern. Each hemoglobin molecule binding 4 oxygens. Each red blood cell being packed with hemoglobin allows a high degree of oxygen carrying capacity. And oxygen travels in the blood bound to hemoglobin. Very little dissolves in the plasma. CO2 is a bit different though. Some dissolves in the plasma, still not a lot, but more than oxygen. By far, the most CO2 is carried as bicarbonate in the blood through that carbonic anhydrase reaction. You ladle in CO2, it disperses to form bicarbonate and hydrogen ions. Some can bind to hemoglobin and or other proteins in the blood, but still a modest percentage do that. 70% travels as bicarbonate. And then other than what we just talked about today, regulation of ventilation, we saw last class that the affinity of hemoglobin for oxygen specifically can change. It's not a fixed sigmoidal curve. You can push it up, you can push it down, and that changes the affinity or how likely hemoglobin is to bind oxygen. And I think we discussed at length how these characteristics are preferentially leveraged at the tissue in a situation like exercise to help with the offloading of oxygen. And that's that.